Hi, my name is Valentina Davila and we are going to present this article, Further Evidence Supporting a Potential Role for ADH1B in Obesity.
So, obesity is a chronic, complex, and multifactorial disease which usually begins in childhood and adolescence, and which has its origin in a genetic and environmental interaction. It's a condition of excess body fat that occurs when a person's BMI is above 30. Our genetic makeup also plays an important part. Studies on families and on separated twins have shown a clear causal hereditary relationship to weight gain. Recent studies have also found a link between obesity and the gene ADH1.
ADH1B is an enzyme that in humans is encoded by the ADH1B gene. The protein encoded by this gene is a member of the alcohol dehydrogenase family. Members of this enzyme family metabolize a wide variety of substrates. The human gene is located on the chromosome 4 in 4q22. This is an enzyme that catalyzes the first step in the oxidative metabolism of ethanol, in which ethanol is oxidized by the acetaldehyde.
So, in the goal of the article, they perform molecular studies using human subcutaneous preadipocyte cell lines to examine the role of ADH1B protein in adipocytes and the potential involvement of ADH1B in the development of obesity.
In this technique, human preadipocytes were taken and cultivated for 14 days until they are shaped and differentiate to adipocytes. The glucose uptake assay protocol is based on a fluorescent glucose analog, which just like glucose can be taken up by cells through glucose transporters. The results show that deficiency in ADH1B protein expressed significantly reduced adipocyte glucose uptake following insulin treatment by almost 40% compared to control.
So, eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of a cell, these mechanisms need to be precisely targeted. There is a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. It's a process in which RNA molecules inhibit gene expression or translation by neutralizing targeted mRNA molecules. There are two types of small RNA that are microRNA and small interfering RNA, known as siRNA, are central to RNA interference. These small RNAs can direct enzyme complexes to degrade mRNA molecules and thus decrease their activity by preventing translation or post-transcriptional gene silencing. So, what do we use it for? So, in the article, knockdown of ADH1B was performed by transfection of differentiation preadipocytes with ADH1B, a specific pool siRNA or not targeting control siRNA. qPCR is one of the most important techniques these days in biomedical research. qPCR can be used for quantitative gene expression analysis, copy number analysis, and differential gene expression analysis, and also variety of diagnostic uses now. It's a technique that is able to amplify and detect changes in the amplicon concentration. qPCR collects data during amplification by using fluorescence signal emitted by special proofs or DNA binding dyes. Real-time polymerase chain reaction (PCR) is the ability to monitor the progress of the PCR as it occurs in real time.
The expression of the ADH1B only starts to show from day 2 and is directly proportional to the days. The expression of beta-actin is always present because it is a protein that is used as a test control in all cell lines. ADH1A and ADH1C, they are not expressed because they have a higher concentration in the liver than in the adipocyte tissue. Also, we find that DUT4 is always expressed in the tissue. And PEPA receptors have a double expression because we see a double line from day 2 to day 14. Protein and genetic material were evaluated in this graph. You can see the increased expression of ADH1B in the MYC, and the FAP4 has a decreased expression.
So in this result, we can see that insulin promotes expression of ADH1B in lean and obese adipocytes. At a higher insulin concentration, we have a higher expression of ADH1B. The actin is the control protein, so they took cell lysates from lean and obese adipocytes, following treatment with increasing doses of insulin. Also, after the treatment with an insulin concentration of 0.05, it revealed an increase of approximately twofold in the expression of ADH1B in both types of cells, obese and lean cells, while treatment with an insulin concentration of 1 produced an increase in 3 or 4 times. So adipocytes were treated with an AKT-specific inhibitor to suppress phosphorylation of AKT to the active form. So we have AKT protein, PAKT, and ADH1B. The AKT signaling pathway promotes growth and survival in response to extracellular signals. So we can see that putting 0.05 micromoles of insulin for AKT was phosphorylated, and ADH1B expression increased in comparison to the control, that is this line. Thus, insulin-stimulated ADH1B expression required AKT phosphorylation, implying AKT kinase is an important regulator of ADH1B in the insulin signaling pathway.
Winner said, our previous and current findings have also revealed that ADH1B gene and protein expression levels are clearly inversely correlated with BMI. De Franco and Prata said, we and others have demonstrated that T2D is an inherited disease characterized by insulin resistance in insulin target tissues, notably adipocyte tissue, skeletal muscle, liver, and pancreatic beta cells. Burke and Dujirala said, we previously showed that obesity, insulin resistance, and T2D traits have a strong genetic basis and high prevalence rates in Mexican Americans, and all these authors were agree with the article.
So to conclude, the expression of the protein and the ADH1B gene is inversely proportional to the BMI. It means that the more the BMI increases, the lower the expression of this gene will be. ADH1B plays a functional role in the cell response to insulin. Insulin-induced ADH1B expression results in a proportional increase in activity and also induced enzyme activity. Also, knockdown of ADH1B expression decreased insulin-stimulated glucose uptake. Also, a higher concentration of insulin shows a higher expression of ADH1B.